I think we could spend a whole week just talking about how we sample and analyze soil organic carbon. So this is just the start of the conversation. And what my objective in this talk is just contextualize a bigger picture. Because I think the Agricultural Climate Solutions program is great. It's getting us started on what we should have been doing 20 years ago, but we need to be doing more. And we can. And I think we'll be able to use this program to really catalyze some important collaboration. So I'm just gonna bombard you with some ideas that are not super well organized and probably will just cause more confusion at this point. But I'm going to talk a little bit more about sampling design, building off of what Kirsten just presented, but thinking about scaling to a bigger picture, landscape and over time, to meet some other objectives that I think we really need to be focused on. Talk a little bit more about laboratory analysis and throw out this idea of trying to develop a hierarchical sampling strategy and leave you with some ideas that you should be considering. So some of the questions beyond the field or plot that I think we need to be driving towards is how, for example, how do changes in the beneficial management practices that we might be trialing, how do they compare broadly with the practices in the region? So we might have a few different treatments in a particular field, but management varies enormously across any particular commodity or region. Importantly, we need to know a baseline. We need to be able to have a good picture of where we stand in terms of soil health, soil organic carbon. And then we need to be able to track that over time. What's the trajectory of that baseline? Something that I think many of us are really interested in figuring out is what is the maximum that we can do in any particular system? How much soil health? How healthy can we get our soils? We don't know. And then where do we prioritize interventions? Where do we get the most bang for our buck across our incredibly large, diverse province? And then how can we efficiently track our impacts? So we have some baseline data. We have soils data that was collected across the province. There's a smattering of it. It's very old. It's got lots of issues in terms of using it as a baseline. We don't, for example, the resolution of what's in this database is too coarse really for us to use to determine any sort of impact on management. Any particular soil that was sampled here doesn't have a GPS location that is really useful for us to determining that space or the time over a particular space. I shared this paper with Shauna that I hope will be distributed. This is a work of a PhD student who graduated a few years ago, Sid Paul, who's working for AAFC now. And we did some analysis as a baseline across the Lower Fraser Valley with funding from the Ministry of Agriculture. And what Sid did was he used satellite imagery, Landsat, to determine land use at 2 points in time. So using satellite imagery to go back to 1984 and then sampled in 2018 and determined land use, land change cover over that time period. So because we don't have that baseline that is so important, we had to use a modeling approach. And basically we came up with soil organic carbon distribution across the valley. And then using that modeling approach with satellite imagery, we determined change over time from 84 to 2018. And what we showed was that on this landscape, this intensive production area, 61% of the landscape was in decline. Only 12% of that landscape was showing an increase. That was using our back of the envelope calculations, almost 6 megatons of carbon. If you convert that to CO2 equivalents, that's That's roughly 10% of Canada's annual emissions from agriculture. So we're talking about a sizable amount, but I would say that this is a hypothesis. This is a modeling exercise. What we need to be doing is tracking this change over time going forward. And so I'm going to show you the different land use changes that we sampled all across the valley and highlighted the annual crop that didn't change was certainly the worst, along with annual forest that got converted to annual crops. And then our best case scenario was perennial crops from annual crops or forest that didn't change. And you can see that the forest that didn't change could be used as sort of that reference case in our landscape, that if we didn't do anything to what was once a forested landscape, that may have been the trajectory. So the scaling methods that we would use in this case, we sampled all across the valley. We sampled in a stratified approach, which I'm going to describe in a second. And we separated these out into training data and to validation data. So we had a total of 310 plots across the landscape. We used what's called conditional Latin hypercube sampling. Fancy term for using a bunch of different data to help us differentiate characteristics of the landscape. So this is showing two approaches for landscape analysis. One where you just sort of sampling in a grid pattern across the landscape. The other is stratified by a bunch of different characteristics. One of the characteristics that we used was you had to be close to a road. You had to be able to get to the site in order to be able to sample it. We stratified it by land use. We needed a certain number of berries. We needed a certain number of vegetables. So using that stratification approach, we then sampled fields where we, within that field, stratified. So you can imagine that the way that we stratified it is a bit like your potential areas for setting up plots. We divided all of our fields into 4 quadrants and then randomly set a point in the middle of that. One of the things that was really important to us was getting good GPS data. So all GPSs are not created equal. So if you're using your iPhone, you're getting 3 to 9 meters resolution. If you're using a Garmin, that's not actually much better than your iPhone. The thing that you buy at MEC is going to get you 3-meter resolution. We were using a differentially corrected GPS that got us sub-meter down to 50 centimeters when we did post-field analysis. So we would set a GPS point, and it takes a long time actually to get a signal sometimes. So one of our strategies was just get these 4 points with the precision GPS, and then we just use a tape measure to get to other points directly from that. So we were trying to minimize the amount of time we're sitting around waiting for satellites. So we use this setup to actually gather 4 different points of soils. So from that GPS point, we were trying to basically get a sense of what a pixel of soil was. So when I say pixel, a Landsat pixel, 30 by 30 meters. So we've got this set up, so we composite those soils. We basically can say that these soils represent 30 by 30. I don't think that's what we want to continue to do. The resolution on satellite imagery has gotten much better. We're now at 10 meters with Sentinel. But I think we can adapt our approach so that we can make sure that what we've done will match with what is planned. So we sampled down to 100 centimeters. We did bulk density only down to 30 centimeters. So I think one thing to really be conscious of is the difference between apparent bulk density and the bulk density that's that we're used to. So I think what Kirsten described would be what we would call apparent bulk density, because when you slam a hydraulic core into the ground, you are going to be causing some change in the density of the soil. Whereas when you dig a hole and then carefully insert a sampler and pull out that core, you're going to be getting a much different type of accuracy on that bulk density. And that's why equivalent soil mass is so important, because you're actually using a parent bulk density to create these calculations. So the other point that I want everyone to recognize is that there are opportunities to sample not just agricultural fields when we talk about soil organic carbon and soil health. One, because there's this idea of what's the maximum, what's our reference point, but also because there are other beneficial management practices out there that we want to be characterizing, including hedgerows or riparian buffers. So we may want to think about strategies for those other important beneficial management practices, or just wildland components of our agricultural system. So we adapted our method, as Kirsten was pointing out. I mean, when we're talking about perennial row crops, we're really talking about two agricultural systems in the same field overlaid on top of each other. So we composited both the row and the drive row into one sample. I don't think we should be doing that. We need to be separating out and understanding the management of the row crop and the drive row differently, because they are managed quite differently across the different farmer fields. And so what I'm suggesting is that we make sure that we're sampling specifically in the drive row and specifically in the row. And this is an illustration of how we did our sampling. We basically had two auger holes. That one was for developing the cumulative mass, or the equivalent soil mass, down to 100 centimeters, and the other was for our composite sampling. And then there were the bulk density samples that we only were able to get down to 30. And some of the locations that we sampled, even getting to 30 for bulk density is really challenging. So I'm not going to go into the spectral or the scaling analysis. Basically, we used a remote sensing method to scale up. I think that's a conversation that we should have later. I do want to, I got 2 minutes. I do want to focus for a second on the laboratory method. So I think many of you know that in my lab, we've got a mid-infrared spectrometer. We use diffuse reflectance approach to characterize the soil properties. What this does is it gives us a way to, I don't know that we're that much faster than Alico doing the combustion method that Kirsten described, but that combustion method gives you carbon and nitrogen, maybe sulfur, and it does it really well, much better than this does, but it only gives you those three. And what we've found is that you can actually characterize a whole suite of soil properties, not at the same resolution that you would get with standard laboratory analysis, but at a much, much lower cost. So I refer you to this paper, a recent paper that was put together. I think many of you know Francesca Cortrufo and her group, or Francisco Calderon. But they've been characterizing, using FTIR, the mid-infrared spectrometry, other types of carbon. So we haven't even talked about the different types of carbon that are out there besides inorganic and organic, but it's not all created equal, and it's all not as recalcitrant or reliable to stay out of the atmosphere as in an equivalent way. So they actually characterized soil organic matter, soil organic carbon, minerally associated organic matter, and particulate organic matter, using the mid-infrared spectrometry and the more reliable laboratory methods, and got really, I think, reasonable relationships for all of these. For us, Our relationships for carbon and nitrogen using the spectrometry approach are way better than what's being illustrated here. I think we have an opportunity that we can develop these spectral libraries. If we run our analysis in Kirsten's lab on the LECO or send it away to ANL or wherever you're using, Dave, have those samples run in a lab. We can start to build the spectral library. Were you guys get up in UNBC getting an FTIR?
In the pipeline.
In the pipeline. So we may have 2 FTIRs in the province, but I think if we build this spectral library, it means that we can run samples much more cost effectively. And then just to illustrate this, I'm not going to go through this in detail, but again, we can share this paper. Paul also was responsible for this analysis. Basically, we illustrated the cost benefit analysis of using lower resolution or less accurate methods like our mid-infrared analysis compared to the standard methods, and just did a cost benefit analysis by using one field as an example. We mapped a bunch of different soil properties across this field, and then just identified sort of the diminishing relationship between the accuracy and the number of samples taken and the costs. And so basically, not to go into the details because I'm out of time, you can sample a lot more with the cheaper method that doesn't give you as much accuracy, or you could sample a lot less with the higher resolution method. So it's something that we want to be thinking about, that maybe what we want is a hierarchical approach where we're sampling across landscapes. We're using both the higher precision accuracy methods for analyzing the soils, and then maybe taking more samples so that we can sample more fields, particularly for the baseline. And if we're able to, and I'm hoping Bing's out there with his, I think Bing has a drone. If we can get drones out there, so we used a drone to capture a 5-meter resolution set of images for this one particular field. If we could use drones to help scale from the plot to the landscape, we can then scale from the field to we go the plot to the field, and then the field to the landscape. We can start to develop an actually cost-effective methodology for monitoring soil health at policy relevant scales across the province. And I think that's what we want to be doing. We want to be doing it so that we can provide management recommendations to farmers based on performance of best management practices or beneficial management practices. We also need this bigger picture approach to see how we're doing overall. So some considerations. When we go out there, if we coordinate successfully, we can get a good baseline analysis for, I think, a lot of our landscapes. If we are doing that, I think we should be considering other land uses. So, maybe that's not part of the Agricultural Climate Solutions program, but those of us who are working on this can be interjecting our other projects so that we're getting this kind of data at the same time. And then consider that trade-off between the accuracy and the cost. And, let's work together to build this provincial or national spectral library. And then if we are coordinated, I do think we can build this hierarchical sampling approach. It's really going to make all of this much more cost effective and important for long-term analysis. And I think that's what we want to be doing. We want to be thinking about what we're going to be sampling in 5, 10, 20 years, because this is a long-term project for us all, for sure. Okay, that's it.
basically in the discussions group, it's a chance to talk about what is going to be the strategy for your projects around soil organic carbon sampling. And are there other things you want to be considering? I know cost is a big constraint on these projects, so I think what you can do in your discussion is you could flag other items, like these are the things we would like to do if we had more resources, because then those could be opportunities for building things on.